I'm a little bit nervous. So let's go on to it. Our theme is rethinking our drug approach. And I put this slide on is the developments and approvals from the FDA in 2014. And as we see that priority review and fast track are about 61 and 41% respectively. This is put just to show that probably congresses like this will be obsolete in the near future as everybody is moving into development of new products using biologics and recombinant proteins, leaving behind the natural chemistry and the mode of developing new products as it was in the past. And we have today that about 19% of the total pharma turnover is run by rare diseases and orphan drugs. The core values used to be the patients, the investors, but nowadays it's with the golden egg that they look for, and the innovation from design mode into a more biotech type of development. Now, the model used to be without the venture capitalist on the far right of the slide, but nowadays risky investors seem to be governing the new products that we see in the market. We have a ton of new biotech companies promising something that is sort of a bit out of this world as the magical treatment for something, and more times we see failures than the development of products that do add a substantial benefit towards the patients. So it's more of having a risky investment, something unique, something that could be potentially highly priced and have a high yield ratio to investment ratio payback rather than what is meaningful. The drug development used to be entities up until today for new chemical entities, identify disease components, possible targets, discover potential leads, then preclinical testing, clinical testing 1 and 2, and then phase 3 clinical trial. And all these do take a substantial amount of money, effort, failures, and these failures tend to be even more now with all the biotech companies coming into play. And further to this development, there is the time needed for the commercialization, scale-up of the manufacturing, registration of the molecules or the products, and then commercialization. And we see that on average development time, it takes about 15 years. Average cost about $1 billion. Now we're approaching maybe 1.5, 1.7 billion of cost for drug development. The patent coverage is about 20 years, and the companies have about 5 years margin to scale up, register products, launch them, be reimbursed by authorities, and make up their profit. That's why we see the statistics, at least, were about 1 in 4 molecules being launched and commercialized being profitable for a company. And furthermore, we see new developments following the evergreen model, trying to either patent an isomer of a certain compound in order to have a patent extension or a relaunch of the same thing through a differentiated chemical in order to get a profit out of the invention. So fixed dose combinations is an alternative mode of developing products, and as per definition, they're there for simplification of therapy, to improve compliance of patients, counteract of one active to the other their side effects, or enhance the activity of each one. So that's a meta-analysis for simplification of therapy, that it does work as a mode of innovation, and not many companies do follow this model to launch new products due to the complicated regimens that are out there. And The development model could go from clinical phase 1 and 2 trials to phase 3 if needed. But in our case, in AVVA, that we're going to talk in a little bit, we do preclinical testing as well, just because we do repurpose certain active ingredients. So AVVA is a company that is research-based pharmaceutical company, and we develop medicines from our knowledge of the human microbiome. And our vision is to simplify medical care by introducing more usable products into the market with either increased safety or increased efficacy. So our background, we all know that the human microbiota starts at birth, and a baby born is sterile by the gut, and then it acquires its biome from lactation and food sources. And it's known that microbiocenosis is an integral part of metabolic processes of humans. And let's go to the products. We have 3 products that I put here just to go over: Lactomezol, Eco-Antibiotics, and Lactofiltrum. H. pylori infection is widespread. It's in fact about 50% of the total population is infected with this pathogen. And we see there is increased resistance and decreased efficacy of our regimens throughout the years just from overusage of these lines, like the Maastricht guidelines, which is 2 antibiotics plus a PPI. So there is a large unmet need in this market in order to limit the patients or have an effective therapy. In AVVA, we saw that through literature that in babies or kids, adolescents, when they're tested, many babies are positive for H. pylori infection. And then due to the guidelines in prohibiting most of the cases to be treated for the H. pylori, and they're retested after a number of years, the H. pylori is not present anymore. So something happens because without pharmacological therapy, the H. pylori is eradicated. And we know that it's a dynamic process within the gut microbiome and the stomach microbiome itself. And looking into this, we examined the concept of competitive inhibition. Whereas in kids that are infected with H. pylori, the biodiversity within the stomach is much less than the biodiversity of a non-H. pylori Helicobacter infected child. So, as we said, the treatment of Helicobacter pylori is a complex therapy, and it's being guided or governed by the Maastricht Consensus Conference or the Kyoto guidelines that take part every year. And Lactomezol is merely a combination of a PPI and a prebiotic. And we're looking into this innate mechanism of competitive inhibition in order to eradicate this pathogen. And these are the results of our interim analysis, and we can see that with our combination, we managed to have close to 60% eradication rate. Comparable to the antibiotic treatment, this might not seem like a good number, but we have to see that the whole healthcare, how we can reshape the attitude of the practitioners, meaning that even if it's 50% eradication rate, it means that 50% of the patients that would need antibiotic therapy would now need it. So antibiotic resistance is not only due to only for H. pylori, but to all pathogens. So this is sort of a holistic approach into how do we deal with antibiotics themselves. And due to the algorithm of treatment of Helicobacter pylori infection, we see that this is a new product that has a repurposed active ingredient, an API, which at low doses, it acts as a prebiotic lactulose. Above certain doses, which are around the 7 grams daily, they have a laxative effect. So this one is an approach of repurposing and including or formulating a fixed dose combination. Eco-Antibiotics. This is an example from the line that we have developed recently. So the background for this, we see too many articles or too much research lately on antibiotics, on biodiversity of the gut microbiome, and the linkage of many diseases to the human gut microbiome. And these are a few examples, 3 examples to be exact, that link the obesity, diabetes, and gut microflora. And it indicates that the use of antibiotics does have an effect on the microbiome diversity, therefore may lead to these metabolic diseases. The role and influence of gut microbiota in pathogenesis and management of obesity and metabolic syndrome. Again, metabolic syndrome, we have fatty liver disease. Metabolism of lipids is affected by the interaction of microbe-host induced microbe-host linkage for the metabolisms. And lately, the last one is a very, very new article that has a link between the gut microbiota and the anti-islet cell autoimmunity, which leads the alteration within the gut microbiota to the development of type 1 diabetes in adolescents. So what we have done in our formulation, we included prebiotic in our formulation of antibiotics. And we have done the DNA sequencing of the feces, and we have found out that if you add a prebiotic within the combination of the amoxicillin clavulanic, coamoxi, you protect the biodiversity of the gut, whereas you have 39, we've identified 39 species being deleted by coamoxi versus 3 with our formulation. And this is distribution of taxonomic groups, and we can see on the left with coamoxi, there is less biodiversity versus the EcoClav group. This is a small trial that was done in pediatric patients. I just put it for the sake of having it. And we have seen that the incidence of diarrhea or loose stools as reported was greatly reduced with the addition and protection of the human gut microbiota. And the last product is for atopic dermatitis, is atopic diseases, means out of place. So it's quite, makes one wonder, why do we use either immunosuppressants or topicals or systemic for atopic diseases, whereas we just, moreover for topicals, we're just treating the manifestation rather than the issue itself. And we all know that the gut-associated lymphoid tissue is the largest immune organ in the human body, maintaining homeostasis and managing different pathogens. So the role of microbiota, as you see, is suppressing overgrowth of pathogens. There is the concept that we talked before of competitive inhibition that is applied for Helicobacter pylori infection. And we know that there's a crosstalk between gut microbiota and host that affects the innate and adaptive immunity. So the Lactofiltrum is a combination of lignin, which is an enterosorbent, and lignin and lactulose. Lactulose, as we said, is a disaccharide. It's an undigestible oligosaccharide. It's being fermented by lactobacilli and bifidobacteria, exerts a bifidogenic effect. Therefore, in patients with atopic diseases, namely atopic dermatitis, we see that we have a compromised gastrointestinal barrier. So our aim through this product is, with lignin, remove the toxins, the allergens within the gastrointestinal tract, and through the induction of the bifidogenic effect of lactulose, reinstate the gastrointestinal barrier and stop the manifestation of atopic dermatitis symptoms. So as we said, most of the products out in the market, they target either antigen expression or depress inflammatory response. We are into reinstating the impaired intestinal barrier function and stabilize it with the Lactofiltrum. Its efficacy, we have quite a few trials that are in combination or in monotherapy in different disease severity cases, and now this product is expected to get within a couple of weeks the CE mark as a medical device. It's a non-systemic acting product.